most people think roads or roadless roads. I'm going to talk about a different infrastructure here tonight, that of an e-science infrastructure, an infrastructure of computers, networks, and storage. And they do what infrastructures do for society. They support scientists in research. And e-science refers to the types of science which make systematic use of advanced computational tools. I work at the Swedish e-Science Research Center, and I'm involved in cancer research. The Swedish e-Science Research Center is a collaboration between 4 major universities in Sweden: Linköping University, Stockholm University, the Royal Institute of Technology, and the Karolinska Institutet. And it was founded on the idea that many fields in science share a need for expertise, competence, and computers. So, I work in cancer research. And cancer is a very diverse collection of diseases. They all involve cells which divide uncontrollably, form tumors, they invade other parts of the human body. So why is cancer so complex? Why is it so complex to define and predict who gets cancer? Well, in the same way that we are all different, we are all unique, every cancer is unique. We all have our genomes, our DNA blueprints that can be used to build an entire human being. In the same way, the tumor has its own genome. So it's very difficult. They behave differently. They respond differently to treatments, and this must be harnessed in order to achieve the personalized healthcare that we're all looking for. And when we want to understand more about cancers and tumors, we need to look at its blueprints. The blueprints is the genome, the DNA. And the process of obtaining the DNA from cells, either human cells or tumor cells, is called sequencing. And for the last 5 years, there's been a technological breakthrough in sequencing technology. In 2000, when the first genome was sequenced, it took 10 years. It cost around $3 billion. Today, we sequence the genome in about a week, and it costs less than $10,000. And this really opens up to do much more advanced science. We can address larger problems. We can look at entire populations and use the genetic information from these. But it also puts a lot of demands on an infrastructure, to an e-science infrastructure, to store and analyze these huge amount of data produced. And we're talking a lot of data. So we say that the sequencing is in the order of terabytes. So how large is a terabyte? It's sort of difficult to grasp these numbers, but you usually say that a terabyte contains 300,000 MP3 songs. 2 terabytes, you can fit an entire academic library in, and so on. And we have several terabytes per experiment usually, or at least study. And we have hundreds of these studies in Sweden. So you can imagine our infrastructure for storing, just for storing these data, is in the order of petabytes, thousands of terabytes. They say 2 petabytes, you can store all academic libraries in the US. And even so, there are some installations in the world where they're storing sequence data in the order of exabytes. I've read that 5 exabytes would cover all spoken words by humans ever. It's a large number. And we don't just need to store this data, we need to analyze it. And we need an infrastructure of tools and software to really transform this huge amount of data into accessible information, accessible knowledge. And the internet fundamentally changed science it fundamentally changed life science as well. We can now collaborate, and we do that on a daily basis all over the world using the web as a medium for sharing ideas, sharing results, and so on. And in the same way, these new technologies, these next-generation sequencing, and other high-throughput technologies, they're fundamentally changing life sciences. We're going towards a data-driven life science world. And this sets a lot of constraints, as we've said, on the infrastructure. You may have heard of the Watson computer. It got famous for beating Jeopardy champions. It's now being applied for cancer research to act as a super library and to sort of translate the vast amount of information in databases to aid doctors when making decisions towards individual patients. It's going to be very interesting to see how this project evolves. And I will spend a minute to talk about studies on healthcare in populations, because these are very important and are cornerstones for public health research. In these studies, we generally follow a group of people. Over time, some will develop cancer. Over time, again, some people will not survive this cancer. And the traditional approach has been to record such information about these people: age, sex, family history, any relatives that have cancer, and so on, and try to make predictions, try to devise risk factors, who gets cancer, who are in a risk group. More recent approaches are to also include genetic information in this. And these studies are large. So there are, of course, some problems when doing this. The first one is time. It takes a long time to make these studies. It takes a long time for people to develop cancer. It takes even longer to be able to measure the outcome of a treatment. Was it successful or not? The second one is size. You need to start with reasonably sized collections of people because not all will develop cancers. And you need to get enough number of cases to be able to make statistically sound conclusions. It also costs a lot. You need entire organizations to handle hundreds of thousands of people followed over years, collecting samples, collecting what happens to these patients. And it's also very difficult to get the control group. As soon as we encounter a case of cancer, we treat it. To have a control group of untreated cancer, that's just not going to happen. And finally, the decisions that you make during these studies are final. You cannot restart a study, do it all over from the beginning, and change the screening intervals. It's impossible to do that. You have to start a new study from the beginning, and it would take even longer. So is there a way we can attack this? So I will describe a recent initiative at the Swedish e-Science Research Center, where we apply this e-science infrastructure. And what we will do is that we will simulate a population of people. We will simulate them with enough detail, genetic information, and we will follow them over time in the computer, apply predictive models that mimic cancer. And with this, we can see who develops cancer. We can try different hypotheses of treatments, and we can see which one were successful and which one were not. The difficult part of this is, of course, to build these models that mimic cancer. And for this, we've assembled an interdisciplinary team of physicians, oncologists, statisticians, bioinformaticians, and so on, that will work hard towards this end. We don't know yet where this project will end. It's recently started, but we do know that this will give a revolutionary view of cancer. This is going to be extremely interesting to follow. And I would like to just to conclude with saying that life is complex, and diseases are complex. And e-science and advanced computational systems are already a key part of scientific work. And it will continue to be so even more in the future. Thank you very much.